we need to talk about cancer in the cell. Since I skipped stuff from 10.3, this will go a little smooth because we're going to skip some stuff because it was part of 10.3 that carried over. Anyways, cancer is the term that is used to define many different diseases. However, they're umbrellaed together because they're all caused by uncontrolled cell growth. It doesn't matter what kind of cancer we're talking about, it's all because the body cannot regulate the cell cycle anymore. Some DNA mutations can evade the checkpoints in the cell cycle. They get pushed through. So therefore, those mutations will get passed on to the daughter cells, and then when the daughter cells reproduce, those mutations will still get passed on. So these mutations are referred to as gene mutations, which are changes in DNA sequences that code for a gene. Cancer occurs when these gene mutations produce a faulty protein that play a key role in cell reproduction. So we'll talk about some of these mutations, I guess. So as these mutations build up over time, it allows bigger mistakes to happen more easily. It kind of, each one of these little ones kind of chips away at the armor until finally big mistakes can happen. Uncorrected mistakes can then be passed on to daughter cells. The mistakes are more amplified with each new generation. As more mistakes accumulate, the cell cycle increases in speed to reproduce the errors more quickly. It's kind of like the snowball effect. Once you have one little bad thing happen, then another bad thing happens, and then another bad thing happens, and it just keeps going quite literally till it's out of control. So uncontrolled growth of mutated cells outpaces the growth of normal cells in an area. So then we get what we call a tumor, which is a mass of these abnormal or mutated cells. Oma is the Latin, Greek, some kind of fancy word for tumor. That could help you with your parts of a word when we look at new words. So proto-oncogenes are normal genes that when they get mutated become oncogenes. So these are good guys. We like them when they're working properly. These genes code for positive cell cycle regulators. These make sure things are going as they need to. The positive cell regulators plug in and allow the cell cycle to continue. Oncogenes are a mutated version of a normal gene involved in positive regulation. This is the origin of replication error, is these oncogenes. A lot of times, if you have cancer, your doctor is called an oncologist. This on definitely is indicative of cancer when you see it in a word. Okay? Tumor suppressor genes are then the negative feedback or the negative inhibition. These are segments of DNA that code for regulatory proteins that prevent the cell from undergoing uncontrolled cell division. And there are 3 genes, tumor suppressor genes, that have been studied a lot: the RB gene, P53, and P21. These are the best understood tumor suppressor genes. These 3 genes put up a roadblock to the cell cycle progression until certain events are complete. So the roadblock is put up until everything on the checklist gets done, and it gets done correctly. If there are issues with these tumor suppressor genes, that roadblock doesn't get put up. And so incomplete or faulty proteins are getting passed through. These cells that carry mutated genes form a negative regulator, and they might not be able to stop the cell cycle if there's a problem.
So it only...
So P53 is one of those fairly well understood tumor suppressor genes. Mutated P53 genes have been identified in more than 50% of all human tumor cells. And it's kind of not surprising because it plays a role in the G1 checkpoint. So that's the first checkpoint that makes sure before DNA synthesis happens that there's enough energy, there's enough quantities of proteins available. Everything that the cell needs is there. So faulty p53 genes may fail to detect errors in genomic DNA. So if there's a problem with the DNA, it might not notice that before it goes on to the replication phase. So if there's problems with the DNA, that problematic DNA is going to be then replicated. Even if a partially functional p53 does identify the mutations, it may not be able to send the necessary signals to fix it. So, like, there's nothing worse than knowing something bad's going to happen, but then not being able to do anything about it. So that's like a partially functional p53 gene. So the damaged DNA is uncorrected because it can't get the fix there. A functional p53 gene will deem the cell unsalvageable and trigger something that's called apoptosis, which is a fun word, and everybody should just memorize what it means. It means automatic cell death. Like the cell just pops. So if that p53 gene is faulty and not functioning correctly, it cannot trigger apoptosis. Even if it knows something's wrong, it might not have the equipment to say, hey, this cell needs to die before it passes on this bad stuff. So then the error gets passed on to the daughter cells. The daughter cells then each replicate this faulty gene, and it just spins that cycle out of control.